So you made it to the final lecture in AMP1 lab. This one's going to cover the eye and the ear. And behind me over my shoulder you can see this is the same model right here for the eye. This looks very similar to the model that you have in your textbook. So we're going to cover this one in a lot of detail just so you know for 100% of what you expect from the eye and the ear models. Let's get started. So vision. So vision is how you perceive stuff from outside, and then how does your brain use chemical signals to say that this is this color, this is this shape, this is what's happening here. And all you're doing is you're interpreting wavelengths of light. And the wavelengths of light that we can see is anything above 400 and anything below 750. So what we see as light, we only see a very, very, very narrow wavelength of light because we only have 3 different cones that absorb different wavelengths within that narrow range of 400 to 750. Anything below 400 is going to be ultraviolet. Anything above 750 is going to be infrared. And that's a different spectrum. So different animals with different types of chemicals in their visual receptors, they can perceive those other types of wavelengths. But unfortunately for us, we only have 3 different types of proteins that we make, and those proteins only act to 3 different wavelengths of light, 3 ranges of wavelengths of light. And another thing to kind of pay attention. I know this isn't in red, so I know it's not going to be on the final, but this is something that you could be asked in your lecture class, and because we have the time for this, I want to spend some time and talk about it. Have you ever seen a rainbow? If you've ever seen a rainbow, you know that it's red, orange, yellow. So it's red, orange, yellow. Can't really see the yellow, put it there. And it's red, orange, yellow, or ROY, G for green, and then BIV, blue, indigo, and then violet. So ROY, G, BIV. And if you ever think, red has a higher wavelength, which means it bends less. So the refraction of light, it bends a lot less. So if you've ever seen a rainbow, red's always on top. Purple, because it has such a short wavelength, bends very, very, very short. So red is always going to be on top, blue, indigo, violet is always going to be on the bottom. And it's all because of the wavelengths, and it's all because it bends a lot more because its wavelength is a lot less. So hopefully that kind of helps you a little bit and kind of understand how does wavelengths work a little bit, but without getting into the physics. So stuff you need to know. Eyelids are just called palpebrae. And the reason why this is important, eyelids help you clean out all the junk in your eye. We do have other types of things. The eyelashes keep the debris out. The eyelids blink. You have lacrimal glands laterally that when you blink, they're going to take it to the lacrimal duct medially, and that's how you're going to keep constantly making sure that you keep your eyes moist and you keep them clean. You have another protective layer on top of that. You have an area called the conjunctiva. And the conjunctiva in most of the models that I'm going to show you are not there, but it's important to know because this is the layer that first gets infected. And all of us has heard of conjunctivitis at one time or another, and conjunctivitis is just that infection with either viral or bacterial conjunctivitis that makes your conjunctiva red, or some people just call it pink eye. So here's that lacrimal gland that I talked about. That's lateral, and when you tear up, the tears go medially, and they go through this lacrimal duct and then down this duct and into the back of your throat. That's why all of us at one point in our life have said our tears are salty. Either they've gone down the back of your throat into your into the back of your tongue where you do have some salt taste buds, and you can taste them as salty. Some of you lick tears off your face. But this is how you're going to keep your eyes clean. And this is the model I kind of mentioned before, where I said that a lot of the structures from this model are going to be on your test. And the model that I used in class is this one right here. So this is the model we use in class. This is the model that I have the picture of, and this is the model that I expect you to memorize backwards and forward. In fact, you should know everything about that model because that's the model that I'm going to ask on the test, but I can also ask anything on here. And like most of these models, where do I start? There's a lot of things in red, and what's the easiest way to start and memorize all this material? And for the eye, because we're using light, why don't we start with where does light hit first? So the light is going to hit the cornea. And right behind the cornea is an anterior chamber. It's not in red, but I just want to say that there's a chamber there with fluid. Behind that, you have your iris. And your iris is going to be the colored portion of the eye. And in my case, I have brown eyes, so I have a brown iris. That space in the middle is called the pupil. The pupil can get bigger to let more light in, or smaller to let less light in. And if you've ever been to the movies in the middle of the day, you walk out of the movie theater and you're just blinded by the light, it's because your eyes have been adjusted to dark settings. When you walk outside, it's bright. So your pupil is very, very, very dilated. It's very big. And then when you let in all that light hits the back of your retina, and then it kind of bleaches out the photoreceptors. So you kind of go blind a little bit. But right after light goes through your pupil, it's going to hit the lens. And the lens is going to be held in there by suspensory ligaments, which are going to be held in by ciliary bodies. And the easiest way to do this, and how quickly does your eye accommodate to near and far? Look at a pen, look far away. Look at a pen, look far away. And you can see how quickly the brain is going to tell the ciliary bodies to pull those suspensory ligaments. And when they pull those suspensory ligaments, they make the lens flatter. When they relax them, they make the lens fatter. And all that's happening there is allowing this lens to focus the light on the retina layer. After you get through the lens, you have the vitreous humor, which is the vitreous body. If you get to do the cow eye dissection, you come out and all this clear jelly-like stuff comes out. That's going to be the vitreous humor. And the vitreous humor is important because it wants to keep the shape of the eye. It doesn't want the eye to be impacted, because if you deform the eyeball in any way, then you're going to stop the light from hitting the retina. Once you do that, your vision gets very blurry. You have three layers of the eye. Sclera is the connective tissue that's white. Choroid is going to be the middle layer, and choroid is going to have a lot of blood vessels. It's going to be brown because right in front of it is going to be the retinal pigmented epithelial cells that are going to absorb stray photons that are hitting the retina. So this is going to get rid of a lot of debris and a lot of built-up stuff from your retina. And the retina is where your rods and cones are going to be, your bipolar cells, your ganglion cells that are eventually going to be the output neurons of your eye. And then the retina is where all the visual components are going to be. And then once they make sense of that signal, the color and everything else, they're going to send a signal or an axon down to the brain via the optic nerve. And unfortunately, right where the optic nerve is started, in the back of the eye, there's a blind spot. And there's a blind spot there because there's no rods and cones. So that is why this area is called your blind spot. And if you've looked, we've covered everything, except for this right here. This right here, I like to think of it as a dartboard and the center of a dartboard. The dartboard is called your macula lutea. If you've ever heard of macular degeneration, the macular region of the retina is where you have the most cones. So whenever you turn your head from side to side, you keep turning your head because you want your eyes to start focusing on different parts. So the photons or the light is going to hit the macula. You get the most high definition or high resolution information by focusing that photons of light on your macula, and the center of that is your fovea centralis. And this is kind of everything. So whenever you talk about optical components, it's anything that light goes through. So cornea, aqueous humor, lens, vitreous body, light's passing through all that. So they're all optical components. Not necessarily for the lab, because all I want you to know, what are the structures if I pointed them out? And this is just a close-up look of the front, where you have the cornea, anterior chamber, iris, pupil, and then the lens and the ciliary body and the suspensory ligaments. And this is how that fluid is kind of be moved around in the anterior chamber. Here's the model again, where you see the cornea, you see the iris, the pupil, the lens, ciliary bodies up here, suspensory ligaments are there. And then here you can see this is where the vitreous body is. And if you look right back here, and if you pay close attention to the lecture and to the videos I have, they're going to be a lot better than what I'm going to do right now. So right now, if you looked at the back of the eye, you can see there are these numbers. And there are these numbers right there. One of them is going to be pointing to the macula lutea. One of them is pointing to the fovea centralis. If you look, the one that's pointing to the whole thing, where my finger is, that's the macula lutea. Where my fingernail is pointing, that's your fovea centralis. So I won't be able to show you a good view of it because this is a small camera view, but on the video that I have for the class, it will go through all of those structures in a lot of detail. We have many different models for this. We have these, they have different muscles. You can see the iris, you can see the lacrimal gland here, and then you can see that's going to be medial over here where the nasal bone is. You can see that's the pupil, iris, and then they have these muscles. Well, these muscles are just as important as all the other parts of the eye. And here is the model you'll see for the muscles in the Gardens campus. And you have 6 muscles to memorize. 4 are going to be rectus, 2 are going to be oblique. You have one oblique on top called superior oblique. The superior oblique goes through a trochlea. And if you look right here, there's a little thing right there called the trochlea, where you can see the superior oblique muscle goes up and around, and then when it kind of comes down right there, it's going back that way. So your superior oblique loops up and around through the trochlea. Inferior oblique is just on the bottom. So inferior oblique is really easy. It's just the one on the bottom right there. For the rest of them, superior and inferior oblique aren't too bad. The rest of them are so much easier. You just need to know top, bottom, medial, lateral, because they're all rectus muscle. Rectus muscles go front to back, so superior rectus, lateral rectus, inferior rectus, medial rectus. Because this is the top, superior rectus, lateral rectus, medial rectus, and then inferior rectus will be back down there. Another view is this one, where you have the superior rectus, superior oblique going through the trochlea, medial rectus, lateral rectus, inferior oblique muscle. And in your lecture class, you're going to know, or you're going to have to memorize, what are the cranial nerves that are important for the eye movement. And you might say, I'm just going to be a nurse. I'm never going to have to memorize this. As a nurse, you're going to have to diagnose this, because at one point, you're going to have somebody follow a pen. And the reason why you do that, and you make the eyes track a pen, you can see if these different cranial nerves are all working properly. You can also see if the pupil and the iris is going to be changing, because if that's happening, that can be another type of brain injury and any type of damage to a cranial nerve where maybe it just doesn't abduce. Maybe your eye doesn't go out to the side, where one of them does but the other one doesn't. It can be something on a different side or same side that's being damaged. Maybe you have the beginning of a stroke or something like that. So knowing the eye movements are very important for diagnosing brain injuries. We're not going to be doing the cow eye dissection virtually because I don't have a video for it, but really all you're looking for, what does the lens look like? What does the retina look like? Trying to cut through the sclera and the muscles, that's kind of what you're looking at when you're doing this type of dissection in the lab. Now let's get to the ear. So the ear is both hearing and equilibrium. So knowing about what hearing part is and what equilibrium part is, it's kind of nice to keep them separated so you can kind of understand the vestibular nerve and then the cochlear nerve. Because it is the cranial nerve, vestibular cochlear nerve, but the vestibule is going to be equilibrium. Cochlea is going to be the hearing part. So it's important to kind of know how do those work. So on that last slide, I talked about vibrating air particles. Pretty much what the helix and the auricle does, the ear, this is the lobe down there, it's going to bring in sound and it's going to amplify it. That's why your ear is that shape. All of those little curves just brings in the sound and then amplifies it into this tube. That's called the auditory tube. And then all those sound waves do, they vibrate the tympanic membrane. And that vibration of the tympanic membrane will then move the malleus, incus, and then the stapes. The stapes then starts doing little dots on this place called the oval window. The oval window then sends that signal down the cochlea, and that is why the cochlea is involved in hearing. You have 3 semicircular ducts or semicircular canals up here, and because there's 3, you have 3 different ways to turn your head, and that allows your brain to know where is your head in relation to your body. It might sound weird, but if you closed your eyes and you just kind of moved around the room, your brain would know within a few feet of where you are depending on how much you moved. That is why when this nerve comes out, it's the vestibular nerve and then the cochlear nerve. Now let's take a closer look. So here's your helix. The antihelix is right there. You have the external acoustic meatus, which brings into the auditory tube. The lobule is down there, which I mentioned. Then you have the auditory tube bringing it into the tympanic membrane. You have the malleus, incus, and stapes, and then the stapes covers up the oval window. The oval window then sends the fluid and the pressure down the cochlea, which then takes it to the cochlear nerve. And then you have the 3 semicircular ducts, which then leads to the vestibule, which then leads to the vestibular nerve. The only other thing, you have an auditory tube, and the auditory tube is if you've ever been diving or on a plane and you had to pop your nose or pop your ear, all you're doing is this is connected to your nasal cavity and your nasal sinuses, where you can equalize this air that's inside here to the air that's pushing it out. So if you've ever been on a plane and your sinuses are a little clogged up, this air is going to be at a different pressure than inside, and it can start to hurt. But if you just push air back up in there, you can equalize it. So when you're looking at the middle ear is going to be the eardrum, and it's going to be the bones. And if you're sitting there and you're like, well, what are the bones? What are these ossicles actually, what are they? These are real size ossicles for a human ear. And you have your malleus, incus, and stapes. Sometimes they're called the hammer, anvil, and stirrup. I never really see the hammer, anvil, and stirrup, but the malleus is the first one, incus is in the middle, and the stapes is the one touching the oval window. So an easy way to think from tympanic membrane to the oval window, MIS, malleus, incus, and stapes. And that takes us to the inner ear, where you have your semicircular canals. That all brings into the vestibule, which goes to the vestibular nerve. And the cochlea, again, is going to be that area where it's going to be hearing. The way it does it, it's going to take mechanical energy and make it into fluid. And what it does is there's these little hair cells on the membrane that when they get tipped over, they allow ions to flow in, and that movement of ions in the basilar membrane allows you to interpret different frequencies and loudness via the amount of ions that change in your cochlea. When you're doing actual stuff for turning from side to side, this is a good way to kind of think. If you had a bucket of water, and you move it real quick. If you move that bucket of water real quick, what happens is the water wants to stay still. So if you're moving your head and you're moving these semicircular ducts, what's actually happening is fluid inside them will stay stationary. But the tissue and the membrane is going to move. And when that tissue doesn't move, it's going to push on top of these gates and allow them to open and allow potassium to go in. And that potassium going in is going to change the membrane potential, and it's going to allow your brain to interpret that as movement. So you can turn your head from side to side, front and back, side to side, and then you can add any other rotation, and those 3 different semicircular ducts can allow you to interpret where in space is your head. So this right here for sound, between loudness and pitch. Loudness is amplitude, and this is going to be how much do those vibrations occur. But pitch is going to be which part of the basilar membrane is it going to be vibrating. Because if it's low pitch, it's going to go a different part. If it's high pitch, shorter frequency, it's going to do a different part. And you're going to learn more about that when you go over this material in your lecture class. And here's just another look at the innervation of the ear that shows which one does equilibrium, which one does balance. But pretty much I want to spend this time just to look at these models. These are the ear models where you have the helix, you have the auditory tube, you have the tympanic membrane going in right there, and then you have the malleus, incus, and stapes. And it's important when you do malleus, incus, and stapes, you go from the tympanic membrane to your oval window. You have the vestibular and the cochlea, and I can't really show you these really well on these models because they're very small. But you can see the tympanic membrane, you can see the malleus, incus, and then the stapes right there. So malleus is touching the tympanic membrane, incus is going to be right on top of it, and then they both go and they touch. Where is it? They're going to go right there and they're going to touch the oval window. Here are the semicircular ducts with the vestibule. Here is the cochlea and the cochlear nerve. So we do have a couple models for these. They have the big one right here. And pretty much when you are reviewing this and you're going over all this, make sure you spend some time on each different model so you'll understand the different structures you need to know. And that concludes every single thing that can show up on your final for A&P1 Lab. Good luck, and this semester is almost over.